Okay guys, this is Biology, Unit 6, Molecular Genetics, and this is Segment 1 Notes. With molecular genetics, we're going to be looking at the structure of DNA and how DNA codes for proteins and how proteins make up you and I, our bodies. So your DNA is your code and it starts there. So Segment 1 is looking at DNA structure. I just want to remind you, since this is starting another semester, I just want to remind you on the method that I highly recommend that you take your notes in. If this is your piece of paper right here, okay, you're going to draw a line, a margin that is going to include little cues to help trigger your memory. So the main ideas, any quick questions or small diagrams that you might want to include here, okay? Inside the main body is going to be what's on the slides. So this is where you're going to record the actual words there. You can also include some diagrams, certainly, with some of the pictures that you see. Labeling them is nice too. And then at the very bottom, you can do this later. Leave this section open, and later as you review your notes, you can fill in this with the main ideas too. This is going to be a way that you can jog your memory there. All right, so who discovered the structure of DNA? Watson and Crick, a couple guys. Watson was an American biologist and Francis Crick was a British physicist. They were working together and they spent many hours building and rebuilding three-dimensional models of DNA using cardboard and wire. And they were really struggling on how it had to be set up. Around the same time was Rosalind Franklin, and she actually was using X-ray technology to image highly concentrated chunks of DNA. And this is actually one of the images that she obtained. Her image got in the hands of Watson and Crick, and they spent time using what they were building in their three-dimensional models and their understanding on how that had to work, and looking at her picture, it started to click. So they went back and they reworked their models and they came up with the structure of DNA, and it is called a double helix, and you need to know that. So Watson and Crick are the ones credited with coming up with the structure or model of DNA. We all know that Rosalind Franklin played a role in it with her X-ray photo as well. But for the test, Watson and Crick. What exactly does DNA stand for? It's an acronym. Deoxyribo, okay, that's the D part, deoxyribo, nucleic, with an N, acid, okay, DNA. Deoxyribo is a sugar, okay, and then there's the nucleic acid, and you guys remember the nucleic acids was one of the 4 macromolecules that we learned. So nucleic acids are the macromolecule, nucleotides, remember, are the monomer for that macromolecule. So what is a nucleotide? So DNA is a long polymer of the monomer nucleotides. That's really what it is. So the nucleotide is defined as the molecule that makes up DNA, and this is an example of what the chemistry looks like. Okay, do you have to know the chemistry, all these little bonds and everything? No, you do not. But you do need to know that there's a phosphate part connected to a sugar. This happens to be deoxyribose sugar. It's DNA. If it's RNA, it's ribose sugar. But DNA, deoxyribose sugar. And then there's a nitrogen base. See how the nitrogen's there? You're going to see nitrogenous base in your reading. And that's what this is referring to as the base. So please, in your notes, draw a little section, a little box for phosphate connected to right here. You don't have to draw all these letters and everything, just I would make boxes. This box is your deoxyribose sugar, and this box is your base. Now, what does that look like in the DNA? So here is your base right there. Here's your sugar, and here's your phosphate. And see how they link together? Here's phosphate, here's sugar, here's base. Phosphate, sugar, base. Phosphate, sugar, base. This is your nucleotide. A single one of these is your nucleotide. Look at though. There's 2 sides of this DNA strand. So here's another nucleotide. Here's another one. Guys, these are the monomers. They link together to form the polymer of DNA. Okay, so that's kind of how it looks. Small little parts are called the nucleotides. The three parts of the nucleotide, and you've already drawn this in your notes. If you haven't yet, you should. Here's your deoxyribose sugar. What is this little green thing? The phosphate, you're right. And then what are these? A, G, T, C, G, A, C, T, C. What are these like boxes on the inside? Those are the nitrogen bases. You'll also see the word nitrogenous. That's the same thing as nitrogen. It contains nitrogen. So anyway, these dotted lines you'll learn later are hydrogen bonds. Actually, you should write that in your notes. I don't think I have that in the PowerPoint. The nitrogen bases are bonded together by hydrogen bonds. Hydrogen bonds, that's what these dotted lines are. So there's 2 hydrogen bonds between the A and the T, and there's 3 hydrogen bonds between the G and the C nitrogen base. There's only 4 nitrogen bases total. Here's your structure of the DNA. Let's make sense of what this image is. I pulled it from offline, and I really liked it. So guys, what do we call this unit right here? This A and the deoxyribose sugar and the phosphate. 3, 2, 1, nucleotide. This is a nucleotide. Linking them together, now we're starting to build our polymer. This is your backbone of the side. If we refer to the DNA as a ladder, these are the sides of the ladder. So your sugar and your phosphate make up the backbone or sides of the ladder. And then the rungs of the ladder are made up of the nitrogen bases, A, T, C, and G. So if we take this little chunk right here, okay, and we go whoop, and we zoom into it, we can see that there's 2 sides to the ladder. There's the sugar-phosphate groups all making up the sides of the ladder, and then the nitrogen bases are here, and then you've got hydrogen bonds holding the rungs together, okay, holding those bonds. And then the DNA, remember Watson and Crick discovered that it was a double helix structure, and so you can see that this is that double helix kind of structure. Okay? 4 nitrogen bases are adenine, A, thymine, T, guanine, G, and cytosine, C. And you can see them here in their structures. So you got G, guanine, bonds with C, T always bonds with A, C always bonds with G. So you're always going to see these combinations and nothing. You will never see an A bonded with a C or a T bonded with a G. It's always thymine bonded with adenine. You can see the backbone structure made up of the sugar and the phosphate, and then the nitrogen bases are what's making up the middle. Hydrogen bonds are found between them, and then one single chunk of this is called a nucleotide. It's got the sugar, the phosphate, and the nitrogen base. It's called a nucleotide. All right, base pairing, this is the rules. So what pairs with what? Okay, hydrogen bonds are going to form between these 2 base pairs. So adenine will always bond with thymine. Guanine will always bond with cytosine. So you can see that that is the case here too. I'm trying to show you a lot of different ways, illustrations that you can see in DNA. So you got your sugar base. I'm sorry, your sugar is here, your phosphate is here, they're bonded together. Then you have this whole little unit called a nucleotide, which is bonded to another nucleotide, okay, according to the base pairing rules. G will only bond with C. That's the only way it will work. A will only bond with T. Watson and Crick came up with the double helix structure, so it's 2 strands of nucleotides wrapped around each other. So it forms this twisted ladder. I mean, double helix, you can write in your notes, twisted ladder. It's like taking a ladder and giving it a twist. Reminds me of like licorice or something, I don't know. Here's another illustration. So what does this blue line represent? If you said sugar and phosphate, you are right. And what are the rungs, these rungs made up of? If you said nitrogen bases, you are right. And what is this box right here encompassing? It's got a sugar, a phosphate, and a nitrogen base. What is this little tiny part called? If you said nucleotide, you are right. And what do these dotted lines represent that are bonding these together? If you said hydrogen bonds, you're right. I don't mean to be sassy, but you do need to know those questions, okay? So here's a quick review. What's the shape of the DNA called? Answer is double helix. What are the 4 nitrogen bases? Answer is C, G, and A, and T. So cytosine, guanine, adenine, thymine. Name the 3 parts of a nucleotide. Answer is phosphate, deoxyribose sugar, and the nitrogen base. And there are 4 nitrogen bases. All right, and that's it, you guys. Done with segment 1.